Good afternoon, everyone. Welcome back from the coffee break. I'm here today to talk about the work that my lab does. We're a structural biology group, and we've been studying the structure of viruses for a really long time. And based on our knowledge of viral structure, we are trying to develop nanoparticles that are based on virus-like particles for mucosal delivery. So this is what the lab does. We've been studying viral structure, as I said. We've studied various different platforms, enveloped and non-enveloped viruses. Amongst the non-enveloped viruses, we've studied picornaviruses, hepatitis viruses, and we find that there are several properties that make certain viruses more suitable to be exploited as nanoparticles for mucosal delivery. The main focus, of course, is to try and bypass this problem, which is parenteral administration. Mucosal delivery, as you all know, and I'm sure have been hearing about, has several advantages. The first one is, of course, that most pathogens that infect humans are usually infecting through mucosal surfaces. There are several viruses and bacteria that do that. And of course, there are the logistical issues such as pain and the requirement for disposal cost related with parenteral administration, the need for trained personnel. So we really need to start moving towards mucosal delivery, and in particular oral delivery for vaccines. Interestingly, we are in such a situation where we know a lot about viral structures, and that helps us to design certain platforms that can be used. So that brings me to hepatitis E virus, which is a fecal-orally transmitted non-enveloped RNA virus. And based on the structure of this virus, we are trying to develop virus-like particles that can be orally delivered. In the past, we've studied various enveloped viruses, and we try to shift from the enveloped to the non-enveloped as delivery systems. Hepatitis E virus, as I said, is a non-enveloped RNA virus. It is transmitted through the contamination of dirty water. In developing countries, there are large epidemics that take place. There are 3 open reading frames, of which the open reading frame 2 encodes the capsid protein. And the capsid is basically made of, when expressed in insect cells as a recombinant protein, it folds by self-assembling 60 subunits to form a T is equal to 1 V virus-like particle, or VLP. This VLP, or the monomeric form thereof, has 3 distinct domains: the shell domain, the middle domain, and the protrusion domain. 60 of these subunits come together to form a virus-like particle. Now, our lab has been involved in the elucidation of HEV VLP structures for a really long time. And the first structure that you see on the left, that's from cryo-EM at 22 angstrom resolution. This structure was published in 1999. And we find, and I'll talk about HEV properties in more detail, but we find that the VLP can be really modulated, and its size and various properties can be tinkered with to form these virus-like particles. We have, in the past, we've made larger VLPs, which are T is equal to 3 particles, which can also incorporate DNA or RNA. And this has exciting implications in terms of oral delivery. Also, if the particles that you can see on the right of this large particle are surface modulated. So they have their scaffolds or their centers are they're similar to HEV, but the surface expresses a variety of epitopes that are different and are from a variety of heterologous sources. So coming back to the structure, this is the VLP is, as I said, made of 60 monomers. Each of these monomers is highly specialized in the way it's organized, in the way its tertiary structure is organized, and in the way the quaternary structure comes together. There are 3 domains. Closest to the N-terminus is the shell domain, which links to the middle domain, which can be seen here in the green, and that towards the C-terminal is the P domain or the protrusion domain, which forms surface protrusions on the virus-like particle. The beta barrels that you see here, these are characteristic of these sort of RNA viruses. Also, picornavirus also has these motifs, but HEV is distinct because the connections between these 3 modular domains are made of large, extensive hinge regions. And that gives us a lot of freedom for engineering these particles because the folding and self-assembly of these domains is, in a way, independent of they're independent of each other. So when we modulate the P domain, which forms the surface protrusions, it doesn't affect formation of virus-like particles. So we can do an extensive amount of engineering without altering integrity of the VLP. I'd like to talk a little bit about VLPs as vaccines first. The viruses in general are really well suited as immune modulators for the mammalian immune system because of various properties. They've co-evolved with mammals and other hosts for so many years and have learned to be recognized and evade the immune system. Their size makes them just right for being seen by the immune surveillance, and they have repeated epitopes that are expressed in a very small space, which allows the immune system to pick them up. This is a great inspiration for us because we would try and develop a VLP-based vaccine based off this. And VLPs or virus-like particles have several advantages in this context. One is, of course, that these are non-replicating empty viral shells, which can be modulated and which also, when administered, can elicit strong immune responses. These are really easy to purify and to scale up, and can, as I said, can be engineered extensively. Now, when we look at this information in the context of mucosal vaccines, it becomes particularly relevant for viruses that are fecally transmitted or transmitted enterically, like the HEV. And as I said previously, there is a great ease of administration in mucosal vaccines, and for pathogens that infect through the mucosal surfaces, such as HIV and various bacteria, this acts as the first line of defense. So essentially, when pathogens like these infect, they don't reach the blood, and they are met by the immune surveillance of the gut or of other mucosal surfaces before they can become systemically disseminated. And the human gut, in this context particularly, has a very well-developed immune system. So we took the hepatitis E virus-like particles, and in this study, which was done quite a while ago, we did some challenge protection studies where we administered HEV VLPs to non-human primates, to chimpanzees, and upon challenge with the infectious virus, we found that there was quite a lot of protection. There was infection, but then it was cleared around 3 weeks post-infection. And this was very exciting for us because this encouraged us to try and develop the VLP as a vaccine platform. And I don't say a vaccine, but vaccine platform because of its extensive modularity and modifiability. So essentially, what we did here was to take the HEV capsid protein gene and modify the C-terminal to express the P18 peptide from the third variable loop of HIV envelope, and this allowed for, as you can see, this is kind of a model of the dimeric structure of the VLP, of the VLP dimer, and you can see that the foreign epitope is expressed. We found that despite chimerizing this VLP, there was no difference in the structure of the wild-type VLP and the P18 or HIV-like VLP. And also this formed virus-like particles which had as much stability as the HEV VLPs. So we went ahead and tried to look at the various sites on the surface of the P domain of the HEV VLP that could be modulated to try and insert foreign epitopes. And this figure, it may not be that clear, but there are 5 distinct sites where we could add insertions or allow modifications to chimerize the VLP. We then took this chimeric VLP, which expressed HIV envelope fragment, and injected into mice to try and look at the immune response. And I'm sorry, I misspoke. This was not an injection. This was an oral delivery. So that was the exciting part. So we orally delivered these VLPs, or orally administered these VLPs, and looked for both humoral immune response in terms of secretory IgA and serum IgG and IgA profiles, as well as those that were shed in the feces, as well as cell-mediated immune response. And we found that there was a strong secretory as well as serum immune response to the P18 peptides of HIV after oral administration of chimeric VLPs, both in the intestinal fluids as well as the sera. Interestingly, we also carried out subcutaneous injections parallelly to oral, and we found that the oral administrations were a lot more efficient in generating humoral immune responses. These are assays to study cell-mediated immune response, and we found that chimerized epitopes generated strong cell-mediated immunity in the spleen and in the mesenteric lymph nodes and in the gut, which is essentially the Peyer's patches. The second property, in addition to the modularity of the VLP that makes HEV particularly well suited to try and develop this platform, is the resistance that the VLP has to gut environment. Now, the first question that anybody would ask would be, a virus that is transmitted fecally should, as it reaches the gut, should be when it is exposed to enzymes, digestive enzymes like trypsin or extremes of pH, should disassemble and fall apart, but that doesn't happen. With the HEV VLP, we found that it is highly stable and highly resistant to the action of trypsin. We wanted to see if the chimerized VLP, which expresses the HIV proteins, is also resistant in a similar way. And we found that even after trypsin treatment, the VLPs remained intact. There was a certain amount of proteolysis where, as you can see, when there was trypsin in the milieu, chimerized VLPs formed, there was some amount of proteolysis, and despite this, the VLPs remained intact. So obviously the trypsin cleavage site was closer to the site of insertion of the foreign epitope. And also when these VLPs were incubated without protease inhibitors, we found that there was this sort of a dropout, a slight lowered band, but then VLP integrity remained unchanged despite all of these treatments. And that really is exciting because a lot of different VLP-based platforms that are being developed now don't show this sort of resilience to gut and other environmental conditions. We've studied picornaviruses, and there are systems that are based on alpha viruses as well, but none of these show this extent of resistance. There are the common cold virus, which is the rhinovirus. It binds to its receptors from the immunoglobulin superfamily, and it essentially just falls apart as soon as it uncoats, as soon as it binds to its receptor. But then HEV is different in that sense that it doesn't do that. The third property that makes that is good in the system is something that would be a concern initially, which is that a non-replicating protein-based molecule to be used as a therapeutic or a vaccine agent would potentially have the problem of inducing self-immunity and would therefore become non-reusable. But then when we chimerize the HEV VLP, and this graph on the left shows an assay to study the binding of anti-HEV antibodies, but chimerized VLPs did not bind anti-HEV antibodies, indicating that this was essentially becoming immune blind to the HEV antibodies, which was really exciting. And the figure on the right shows the binding site of the monoclonal antibody that was used to study whether the VLP was still binding to HEV antibodies. The fourth and most exciting property of these VLPs is that they are able to non-specifically encapsulate foreign DNA, and this can be extended to RNA as well. And we've done that in the lab, where we took plasmids encoding HIV GAG and encapsulated in these VLPs. And I'll tell you a little more about how we did that, and tried to look, after oral immunization in mice, we tried to look at the immune response which was generated. Since we're structural biologists, we always come back to trying and understanding how this happens. How does a VLP that is not specifically interacting with DNA, with GAG DNA, how does it encapsulate it? There are some critical interactions that take place at the 5-fold and 3-fold interfaces of the folded VLP, and there is a calcium-dependent salt bridge, which you can see, and this is sort of homologous to other non-enveloped RNA viruses. This allows for certain characteristic positive charges to be present on the inner surface of the VLP, and that binds to non-specifically to DNA. And the way we go about doing this is to disassemble VLPs in the presence of EGTA and DTT reducing agents, incubate with DNA, and in the presence of calcium, keeping in mind that the interaction may be calcium dependent, we allow for reassembly, and these particles can be seen to have encapsulated DNA. So we took these VLPs which had encapsulated HIV GAG DNA and orally administered in mice. We found that both humoral and cell-mediated immune responses were generated against the GAG. And this is really exciting because not only was this GAG DNA being released at the correct site, it was being exposed to the immune system, being presented in the correct context in the context of MHC molecules, and generating an immune response. And these electron micrographs that you can see on the right are of mouse gut epithelium, which is this is immunogold labeling of GAG protein, and we found that this is immunohistochemistry, again showing the same thing. Controls that we used were naked DNA, and we found that there was a strong immune response. And this is also to show subcutaneous versus oral delivery of the GAG encapsulating HEV VLPs. The fourth thing, and which is kind of a general point, but this is a really important point in HEV VLP-based vaccines is that we use a baculovirus-based system to express these, being eukaryotic and being relatively closer to the mammalian system as compared to, say, yeast. It allows for correct folding of the VLPs. We find that the VLPs are secreted into the supernatant, and so they are easy. It's easy to scale up. It's easy to purify. And another important factor is that being based on virus structures, they don't require separate adjuvants in their administration. This shows us how much VLP is produced, and this is most of it is really well folded, and it has been used previously to elucidate the structure, and it is also secreted into the supernatant. The second property that I'd like to point out is that the VLP backbone, being based on a pathogen that's co-evolved with the mammalian system, it is able to activate the innate immune system and thereby prime the immune system to act on the chimerized epitopes to generate a stronger immune response. And I'll go over this for now. We were really excited by the concept of the medicine in drink. Keeping all these properties in mind, we know that the HEV VLP can be designed and formulated to allow for chimerized epitopes as well as for encapsulated DNA or RNA to be produced and administered in the form of drinks, in the form of actually pretty much anything based on their stability. They are stable at acidic pH. They are stable without refrigeration. We were excited by the concept that was published in Time, that if we tune our lifestyle and if we tune therapeutics in such a way that through minor changes, through non-invasive changes, such as bringing in just formulations that are delivered orally, we can really improve and enhance our lifestyles and the human lifespan. And with that, I'd like to finish and thank the team and Dr. Cheng, who's here, and for all his support.